Okay, so unfortunately I can't be at lecture on Thursday. I got double booked. I have to actually be out of town to give a talk at a cancer center that was scheduled months and months ago that kind of slipped by. Um, but what I'm going to do today is it turns out we're a little bit behind anyway. I got a bit confused with the lecture schedule. So today, what we're going to do is I'm going to go through some first some principles or some ideas about drug discovery, and we'll do that, and then I'll follow this up. There's going to be followed up by two short lectures on SAR, which will be posted probably um, be posted either Friday or Saturday. Um, at least one of them Friday, maybe the second one Saturday. So you can view those at your leisure. The notes will also be posted on the Moodle site. Um, one of the key, let's see if we can get oh, let's see, we can get, here we go. Let me get a pen. Um, there are several key concepts in drug discovery. It's a process that's taken a lot of different uh, different avenues and different paths throughout the last 100 years, and we saw a little bit of that earlier on, in which people used plants, and they discovered potential things that could be useful in plants. That could useful things that could be used from bacteria, and we'll see that in a little bit. Um, but the whole process of drug discovery has taken on one of the concepts should be is a much more defined, rational approach that's very scientifically based. Um, it's still it's still an art form in many ways. It's nowhere near as exact as engineering is. For example, if I want to build a bridge, uh, there's all the principles about how to build a bridge. Want to build a building, there's all those principles. Really, it's just a different way of doing the architecture. But the fundamental principles about how you build a building or a bridge will always remain. There's fundamental principles of mechanical engineering that are based on fundamental principles of physics. Chemistry and biology are far more complex than that, um, and so there are a lot of things that are approximations. A lot of things in order for us to be able to um, grapple with the rationale, and so there's a part of discovery and there's also part of design. And you'll find today that those both are intertwined. Lots of times you will be looking for one thing and you find something else. Um, and the successful person who's involved in drug discovery always keeps an open mind about what they're looking for. The unsuccessful has a very closed mind, and unfortunately, a lot of large pharma companies have had um, too many. They their their focus has been on how to industrialize the process and not recognizing the tremendous creativity that's necessary to go into actually drug discovery and drug design. Because there is no, because there we have rules and things, but there's a lot of flexibility within those rules. Now, in terms of some of the key concepts we're going to talk about, uh, one of those is what's referred to as a lead compound. And so a lead compound is a prototype having desired activity, but also other undesirable characteristics. Maybe it may have toxicity. It may hit other. It may have other activities which you don't want, like it kills cancer cells, but also kills um, cells that are in the lining of the gut. It may be not be very soluble. It may have metabolism problems, or you can't take it, formulate it very well. But it has the activity that you want, and it has maybe a potency that you want. And so this is where you sort of begin with a lead compound. Um, Sometimes a lead compound will have all the characteristics you want, and it can become the actual drug. And there are a few cases of that, as we'll actually see. Um, but in a lot of cases that's not the case, particularly in drug design. Leads then are modified by synthesis. Generally, they'll be modified to amplify desired activity and to minimize the or eliminate undesired properties, to make them more water soluble, less prone to metabolism, or more prone to metabolism, whatever it is that you're after. That can have, so there will be a lot, and this can go back and forth. Uh, a drug candidate, so these would advance to what we would call advanced leads, um, more highly defined and more efficacious leads. A drug candidate, a drug candidate is then a compound worthy of extensive biological and pharmacological animal testing, and these are compounds that have a lot of really good characteristics, uh, uh, minimum deleterious characteristics, and these compounds um, might actually end up becoming a drug. In some cases, they'll need just another minor modification in the end. But those are the candidates that will then have extensive what are referred to as preclinical evaluation. So lead compound, lead modified, or advanced lead by synthesis, and drug candidates. It's not correct to call a molecule that does some biological activity a drug. I see this all the time. You see this mainly from people who are not very knowledgeable. You'll see this in seminars and all. People will say, well, I use the drug. It's really a lead compound. It's a compound. It's not a drug. A drug is something that's FDA approved and is actually in human beings or animals. Now, you can discover, um, as in the case with kind of older medicines, is you can do drug discovery without a lead. One of the most famous examples of that is the discovery of penicillin by Sir Alexander Fleming. Now, Fleming is famous for discovering penicillin. In fact, he won the Nobel Prize in 1945 for it. Um, but his observation, he wasn't really looking for it. He actually discovered another, he was a microbiologist and actually discovered another protein called lysozyme, which everybody uses, is part of your actually innate immune system because it can destroy the cell walls of bacteria. And in the process of his wanting to um, um, find other things that would kill bacteria, in his looking, he noticed that bacteria could be lysed by this green mold, and the story goes that this mold had contaminated a dish, a culture dish, and this left a dish. He left the dish on the bench top while on vacation, um, and weather was unseasonably cold. I believe this might have been in Scotland, England. Anyway, it's cold there anyway, even in the summer. Um, and this particular strain was a good strain that happened to light onto this mold happened to light onto this dish. It was a particularly good strain at producing penicillin. And you can see that here. Here's the penicillin colony. If you look at this, here's the colony, and then what you notice here, this is actually sorry, this is the penicillin colony. There's a penicillin colony here, this big blob here, and then here's the bacteria. Here's bacteria, streptococcal colonies, and then you'll notice right in here, there's none, and that's because it's actually excreting a compound that kills the bacteria, lyses the cells, and that compound happened to be penicillin. And this is his famous paper on the bacterial action of cultures of penicillium, and special references therein to the isolation of B influenza. And you could see this worked, and so but the problem is that he couldn't reproduce this. Turns out it was a strain, it was difficult to reproduce. And he couldn't, when he was able to struggle to make the spores, it was very difficult to isolate any significant quantities of the compound, and he had no idea what the structure or what the compound was or what it was. For all he knew, it could be an enzyme, as opposed to a small molecule. But he presented this. At a medical conference, to a lot of yawns. Nobody really paid too much attention to it. He was actually a pretty soft-spoken, rather introverted man, and so it just didn't it just didn't get any sort of notice whatsoever, really. About 10 years later, so this was in, you see here, this was in 1929. So around 10 years later, um, uh, a couple of other scientists, Sir Howard Florey and Ernest Chain, in particular Florey, took a real interest in it because they're we're now getting to the point of 38, 39. You're talking about the war, actually. World War II has started in Europe. And they really wanted to see if they could produce this. Now, the methods for doing it uh, were not known, but they had an idea that if they were able to uh, produce the penicillin and grow it, the really most important thing was the culturing of the penicillin, figuring out how these spores grew. And so what they ended up doing is they couldn't find anybody really in England to take this up in any serious way, and so they ended up going to the United States, and there engaged the Columbia University, and it turns out Pfizer, um, which is a major pharmaceutical company now, but at that time was a small company, and Pfizer was the one who started to invest and see how they could make it. Now it was a real struggle to make this compound, to make the pistol. First they had to they tried to identify what it was, um, um, and then the other thing too is that the compound itself was pretty unstable. So whenever they made it, batches of it, the compound, and there's a reason for that. That reason is because when the structure was finally elucidated in 1944, so this was like 4 or 5 years later, um, by Dorothy Hodgkins, this X-ray structure turned out to contain a unique structure, this beta-lactam molecule. And so this is the natural product. This beta-lactam, as you can see, has a formative ring, is quite unstable. Um, and so if you put it in water, aqueous solution for a week or so, it's going to be hydrolyzed. You're going to get attack. You'll get attack of water. You'll get attack of water hydroxyl group. You know, is going to attack at that highly strained ring, and so this will hydrolyze in water. Um, and so trying to figure out, once they would get it, then it would lose activity. They could make but a few micrograms, a few micrograms of it, and the war is starting to heat up quite a bit. And so discovery of this new natural product was profound, and this led then to later the discovery like of the cephalosporins in the 1960s, and sort of other varieties of these penicillins, and what are referred to as the beta-lactams. And these beta-lactams, the key thing there was that Pfizer developed its first methods for the production, because what they ended up doing is they discovered how to do. There was a proposal uh, that by the scientists there at Pfizer that they undertake using fermentation to do this. Now it was a big risk for Pfizer because Pfizer basically had to take most of their other projects offline, set aside, and for the cause of the war, start to figure out how they could produce penicillin. It was actually a pretty risky thing to do. Through a lot of hard work and effort, they were able to actually scale this up and start to produce really medically useful quantities for war effort. And in fact, the U.S. government basically commanded them to take their methods, and they freely did this. They distributed these to other companies to scale up the production of penicillin. Actually, in the end, the Pfizer group was so good at making penicillin, nobody really was able to make it as good as they did, or the quantity. And they actually ended up, for the war, produced about 90% of the penicillin. The penicillin that was they by the time they were able to, they did it rather quickly, and worked night and day to do it. And at D-Day, there was enough penicillin there to provide for the soldiers. And of course, it saved countless lives. And because one of the biggest reasons for mortality in wars up until then was actually the death from, if you got wounded, you would die from infections. You could linger on for quite a while and then die from infections, or die in if you're in contact with people that have bacterial infections. And so to be able to have a drug what was referred to as a wonder drug, um, uh, to save lives on the battlefield, was just a huge advance. It was a huge advance. It's a huge advance. It's not the first. It's not the first time that the use and practice of use of a medical of a new way of treatment has been one of the products of a war. Um, but this was really a serendipity, a discovery, a notice, a certain an observation that was made by a scientist. This led then to actually to further um, probing for moles and bacteria for antibiotics, and it led to an explosion in the 50s and in the 60s, then of the development of a whole new arrays of different antibiotics that were originated as natural products from um, bacteria and moles and fungi. Uh, so much so that by, I think, the early 70s, people were saying that there's actually in Congress, and that they should stop basically all antibiotic research because we don't need any more antibiotics. Of course, that was turned out to be a really dumb thing to say, because even at that point we knew that resistance was beginning. Um, Now, another way is discovery without a lead is by observations from chemistry that occurs that's unexpected. So in 1955, Roche prepared a series of what are called benzoheptatoxadiamines. These are, and they were looking for potential new tranquilizer drugs. This was actually uh, quite an exploding area, the use of potential drugs um, to control the central nervous system. And the actual structure was found to be a quinazoline 3-oxide. So um, the structure of the actual drug. Now, what turned out is that there were no active compound. They made these compounds, which are these heptatoxins, and looking for compounds that had tranquilizer type of activity, and they could find that. In fact, there were no active compounds, and the project was abandoned. Um, and uh, when they went back, like 2 years later, they were throwing out a bunch of compounds. In fact, no drug company throws out their compounds anymore. Most drug companies have made hundreds of thousands of compounds that are within their vaults that they can go back to, if not millions of compounds that they might be able to go back to see what other activities they have. But what they found is that these compound was, when it was stored in this initial the compound in was stored in DMSO, and it had a little bit of amine in it, that lo and behold, this molecule, which is a diazepam, which is Librem, had really great activity. And so what they did is they retested this, and what they discovered is that in in the course of the couple of years this compound had been sitting around in DMSO, it will mean this unexpected rearrangement had occurred, in which you get a ring expansion. There's a ring expansion, and as a consequence, this compound, which are the benzodiazepines, which we refer to as benzodiazepines now, was found to have activity. And so, in this case, they were looking for a compound. They were looking for a compound, and then had the good sense to actually retest some of their compounds to see if they were still to just retest them to see, and they found the new drug. And this drug became one of the leading tranquilizer drugs in the world. Now, how do you discover leads? Now, you need a bioassay, right? A bioassay is critically important, and you saw that with Fleming's assay, where he had an assay where he could tell that the mold was killing the bacteria, and so you need it's absolutely critical what your bioassay is. And so you need to put a lot of attention to that, because one of the things you'll need to do is you'll need to potentially screen a lot of different compounds and see if they have activity, not just that one compound. It's quite remarkable with Fleming's compound that compound we still use it. There's no modifications at all. Of course, there's a lot of drug resistance to it now. There's also allergic reactions that can occur with it. There's a rationale for that. It has to revolve around the beta-lactam. But for most efforts or campaigns for looking at drug discovery, you're going to be needing a bioassay in which you can screen a lot of compounds and gauge their activity. Because what you're interested in is what's the activity and the relative potency. The activity, remember, all compounds can have activity, but they're not all the same potency. So one way to do that is to use high throughput screens, or HTS, and these can be easy, fast, and sensitive screens. And there's a tremendous amount of effort to do this, to make them to be able to get your results fast, reliably, and reproducibly. Most of them are the screens that are done now. You don't have a dish with a mold on it, or you just add a dish with and then add one compound at a time. Most of them come in large, are automated. These screens are automated, in which you add maybe thousands of compounds in plates after plates after plates, in which you can have a plate which has over 3,000 wells in it, and can put in just very tiny amounts of your compound, and then you can read that by whatever it is that you're looking at, fluorescence, whatever it is that your fluorescence, absorbance, light scattering, whatever it is that you're doing your assay for, enzyme activity, absorbance. And for most drug companies, the goal within 1 or 2 days is to assay 100,000 compounds a day in that assay. So when you think about that, that's that's about 30. What is that? That's 10,000. That's about 30 plates, somewhere around 30 plates, 30 of these 3,000 well plates, and so that would give you 100,000 compounds that were screened. There's a lot of pressure to put on by have assays that are fast, so you don't wait 24 hours or not. But that's actually, I don't consider that a real. That's not a real tremendously important thing, as long as you can get accurate information about the compounds. The one issue with most of these screens, though, is the compounds themselves, because time and time again the compounds that you get are dirty, or they're not pure, or there are things in them when they were synthesized. They thought they were pure, but they've either reacted with oxygen or done a variety of other things. And so there is a tremendous amount of literature out. There's a lot of literature out now today about the different ways that screens can lead you in the wrong direction, give you compounds, potential lead compounds that are not actually leads but are false positives. That's the huge bugaboo about when you're screening all of these compounds, is how many of these are false positives as opposed to real positives. The negatives you can't see. Now, a typical pharma company, this is a bit old now, but um, it's been ramped up quite a bit. It's probably, in 1990 you could screen 200,000 compounds per year. By the mid 90s, about 5 million compounds per year. 2000s, 50 to 100 million compounds could be screened per year. You need to have a lot of money, because even if this assay is 5 cents per assay, that's a lot of money to screen those compounds against your assay. Because the reagents actually for the assay itself can be very expensive. If you have a protein, for example, and you're just looking at binding, the protein itself you got to produce enough for these assays, and the protein could be actually kind of expensive to do, that many assays. In most cases, from what I've seen, for a particular hit or assay in which you're looking at a campaign, you tend to see a high throughput screen assay campaign probably around 100 to 300,000 compounds, somewhere in that ballpark. You don't generally see you don't really see campaigns of 1 million compounds. And those compounds, now people have gotten smarter than in the 1990s. Those compounds that you picked are each individually picked for a variety of different activities, for behavior, solubilities, metabolism, variety of different things like that. And in some cases, a bunch of new ones thrown in there. Now, when you do these screens, what you get are several compounds that would be called maybe 100 compounds, 10 compounds, hits, and those are compounds that elicits a predetermined level of biological activity that you are looking for. Level of activity you're looking for. Now, the one thing that happened was when high throughput screening became really all the rage in the 90s, as you're getting out of the 90s, is that the actual amount of drugs that were coming out, the promise was that you'd get lots more drugs coming out, was actually did not increase, but actually decreased, whereas the amount of money going in to produce these was getting higher and higher, because you've industrialized the drug discovery process. To put it frankly, you have a bunch of MBAs running the operations, and they don't know that, and they don't have any idea about the science. And numbers are, the idea is that if I screen 5 minute compounds, I'm going to find something good in there. Well, if everything you have in 5 minute compounds is crap, you're just going to get crap in, crap out. And so it really matters the intellectual effort. It's what turned out is by the 2000s, it was like, we're in really deep trouble. In a lot of cases, this ended up with a lot of drug companies going bang, actually having to be sold. But the whole idea that there's an art, there's a knowledge, there's a science behind drug discovery, all of a sudden people are saying, that we probably should go back and learn remember the principles that were good in the people had figured out in the 70s and 80s, 60s, 70s, and 80s. But there was no increase in drug approvals, and everybody started to understand that. So just screening lots of compounds doesn't really necessarily get you where you would like to be. But I just want to point out that a hit is not necessarily a lead compound. A lead compound has properties that are reproducible, has some of the properties you're looking for, and is something you can hold your put your hat on, that you might be able to then take forward. A hit is just what it is. It's a hit. It's undefined until it's really exhaustively it's resynthesized and it's retested and it's found to have the properties that you want to become a lead compound. Now, for example, here's an interesting some statistics from um, uh, BMS, um, Bristol Myers Squibb, and the number of compounds that they were producing. And as you can see here, they are producing probably in 1998, they are producing somewhere around, it looks like, maybe about 50,000 compounds a year, something like that, 40 to 50,000 compounds. Um, and as it went by, within 5 years, they're up to producing close to a million, or they're producing, a million compounds per year. And it keeps going up to, kind of plateaus out here, but they make this big bump to about a million compounds per year. And so these are lots of compounds that are being produced. And so how do you evaluate these compounds and which of the compounds are good? And that has been a colossal problem for big pharma to deal with. And in the end, it hasn't really led to actually an increase in the number of compounds. Now, if you're smart about your libraries and your chemical libraries, and what's happened is people are getting quite much smarter about it and the design of those, so that they will give you useful information for the drug design effort. So people are getting smarter, and therefore the approvals are actually going up. Now, other screens are called combinatory enzymes and pathway screening, and that's where if you took, for example, if this is a process from going from this metabolite to this metabolite, and there's these three enzymes that are going to go through that, I could screen molecules, I could screen a molecule against all of these simultaneously by measuring these metabolites. So if I put in here and there's an inhibitor of A and all I see is the starting material, then I know that that it inhibited at this junction. If I see this product, I know it inhibited B. If I see just this product, then I know it inhibited C. And if I see a lot of this product, I know it didn't inhibit any of them. Now, it could inhibit one or more of these, and so it's possible that when you go back, you'll find that it might inhibit A and C at the same time, and so those are also possible. It doesn't bias when you do the combinatorial method, combinatorial enzyme screens like this. It doesn't necessarily bias you as to which enzyme we would like to hit, because in some cases, druggable molecules, that means a compound that can potentially be a drug. It has properties that make it more likely to be a drug. Though those molecules might be better suited for B but not for A. So in these sorts of things, this sort of allows you a certain flexibility to discover. It's always good to have a discovery built into some of these systems to give you that flexibility for discovery. Because discovery is nonlinear. That's what really irks the MBAs. It's a nonlinear process, and it's episodic, and you have jumps, and you have long waits, and you have and it's unpredictable since it's nonlinear. So those are one way. That's one way to do it, is to screen against several enzymes in a pathway. Another one is to screen. Screening is also done by electrospray ionization mass spectrometry, in which you can take compounds, and you have your protein, you have a lot of compounds, or say 3 or 4 compounds simultaneously. So this is in which a way I can screen several compounds against the same protein. I mix those, one of them binds, I separate them, and then when I analyze this by mass, I separate the protein bound from the small molecules. I could put this down size exclusion column. This would trap this, and this would fall through, and I then put that on the mass spec. I would then be able to find this molecule by mass spectrometry. It would have a particular signature, and that would allow me to mass signature, and that would allow me fragmentation. That would allow me to to find things that are specific in a mixture. So one of the ways you can screen is you can take mixtures of compounds and screen them against your target. Um, this is done sometime. This is done a lot of times with plant extracts and other things like that. In the late 90s or 20s, or early 2000s, people were actually taking mixtures of compounds, adding them to the proteins or the targets, and trying to identify things that were in that one in that mixture. For the most part, that's not done. I haven't seen too much of that being done these days, in which each assay is a discrete compound and you have thousands of assays that you're running simultaneously. Now, the other concept are concepts are target-based versus phenotypic screening, and these are basically the whole industry, the whole approach is fixed on one of these, one or both of these. In fact, there's sort of both involved. In some cases, one will. In some cases, and when someone's trying to do a drug discovery, they'll be fixated on one, and in another case, they'll be fixated on another. So, for example, for target-based screening, this is a screen in which is the search and identification of small molecules, peptides, or RNA molecules that inhibitory RNA that bind to a specific protein or nucleic acid. So you have the protein, you know it's a target, what you're looking for, and so you're trying to find molecules that will bind and ultimately alter its biological activity. So a small molecule that inhibits a purified protein, or peptide that's an agonist of an opiate receptor of a GPCR, something like that. You know it, and you have purified, and you know in your assay that whatever your readout, it's just going to hit that. You're just looking at what's going to happen to that protein. It could be a cell-based assay, but for example, an assay in which the receptor is turned on and you see chemoluminescence. And so inhibiting that, that's what's happening. Those are called target-based screens, and if you really know what the target is, then most people will try to build their compound or start screening to determine what potential scaffolds would work against their protein. The problem with that is that the issue with target-based screening is it doesn't allow for discovery. Two, your target may actually not be the best target. Granted, someone's done quite a bit of work, but one of the issues in drug discovery, particularly in pharmaceutical, for design of pharmaceuticals, is what's called whether your target is really a clinical target. In other words, is your target really the target that we need to go after? And there's a lot of targets for a lot of diseases. For example, Alzheimer's, there's beta secretase, amyloid formation. There's been a number of hypotheses that have been developed to do that, and so for that target. And so, but what's come out of that is that there hasn't been any Alzheimer's drugs, because when those drugs have been developed and tested clinically, they have not been efficacious. So the target itself, there's something about the target that is not the correct target, and that means we don't understand the disease very well, and there are other things and we don't understand the side effects or potential ways that biology can overcome whatever it is that we're trying to do. And so that kind of target-based screening kind of puts you in a hole. The effort that's gained quite a bit over the last 5 years in particular are called phenotypic screening. And this is actually a very old screening approach. In fact, what Fleming did is phenotypic screening. He saw a phenotype. A phenotype is the biological activity of a cell or organism. So phenotypic screening is a search and identification of small molecules, peptides, or RNA that alter the biological activity of a cell or organism in some way that you're looking for. For example, it may disrupt the junctions between two cells, cancer cells. It causes them to, or causes the cells to be disrupted. They don't adhere to each other, or promotes it. Or cells that are not able to not able to process glucose. It somehow interferes with metabolism. Cells that are not able to produce a particular virus. There's a viral for HIV infection of T cells. If I add HIV and I'm testing for whether these compounds work, I want to see it doesn't produce the virus. The virus is not produced by the T cells. So those are called phenotypic screens. The one issue there is that if you don't know what the target is, if you haven't really a clue as to what the target is, you see the screen, you have to define what that target is, what the target's going to be, and that can be a long, arduous process. In fact, because in a lot of cases, compounds that cause the phenotype, whatever that is, can have multiple effects on multiple different targets. So they're not cleanly hitting one or two targets. What you're observing may be the result of two or three of its minor action on two or three things, resulting in an overall action. And it can be hard to nail that down, extremely hard to nail that down. So that's kind of the drawback that you have with phenotypic screening. But in phenotypic screening, if you do get compounds that do what you want them to do, and they are safe, that's all really the FDA cares about, is they're safe, and they don't have for the use that they're being put in. You don't necessarily need to don't know every single thing about how the mechanism of your drug's going to work. They certainly had no idea how penicillin worked. It took years. It was decades before they understood exactly how penicillin disrupts the formation of the cell wall of bacteria. So there's this constant sort of pull because in this day and time you can't really do that. You need to know what the target is and see where it is, because that will potentially be predictive of potentially long-term toxicities or toxicities you don't know anything about. The good news is for phenotypic screening is that the technology for identification of targets by high throughput genomics, high throughput proteomics has gotten tremendously better, and it gets better every single year, so that you can identify your targets. There's a much better chance of you being able to identify your targets every year. So those are the two things you'll see almost all drug discovery based projects circle around: target based screening versus phenotypic screening when they're trying to discover new compounds. So I think what I'm going to do is I think I'm going to stop here. I'm going to stop here, and I will pick up on the next lecture, on the next part, when we start talking more about SAR, getting into SAR, structure activity relationships.